So thank you for the opportunity to share the data about our COVID rapid test. Why is so important? Well, although we all have the hope that COVID was going to finish quickly, currently we have around 100 COVID cases every 3 seconds in the world. And someone dies from COVID every 12 seconds. In other words, we are still here and we are going to continue to deal with COVID for a long time. So we need to be better prepared. Why another COVID test? That was the question that always pop up. There are currently 400 tests approved by the FDA. As you see, the majority are molecular tests, and mostly they are trying to simplify the process. They have 88 antibody tests and some of the 34 antigen tests. What is the problem with those existing methods? As you can briefly see here, the biggest problem right now, and this has been growing with the Omicron, is there is a high rate of false results. And this is with the PCR, which is the gold standard. And you can see the reference here in the Hopkins website. And if this is bad, with the rapid test, the situation is worse. And with Omicron, as I told you, the situation has getting worse and worse. And you can see in the right side that has forced the FDA to recall thousands of rapid tests from the market. If we translate those rapid, those false test results, that means 300 million false results in the world. So we are talking about a big problem. Why? Why so many false results? Basically because we have a big gap for diagnosis in this side of the curve. And we also have a big gap for diagnosis and for screening in this area. What is the market need? So we try to answer that question with the I-Corps, and we find out that we have different types of hospitals. We have small and medium hospitals that are in both in the urban or the rural areas. And then we have the large hospital and the federal funded hospitals. These hospitals in the right side are likely to be open 24/7, are likely to have laboratories also open 24/7, have PCR, or have created their own tests to respond to the pandemic. On the other side, which is the majority, they are not running laboratories 24/7. In many cases, they refer the results to other sites. That means outbreaks. That means they need to pay for more money. And every time that they have outbreaks, they have less money. And basically, they have less funds to respond to those problems. Moreover, if you look at the majority or the large proportion of cases that are already hospitalized, they have been diagnosed clinically. 30 to 50%. It depends on where you look at it. The numbers can go even higher in developing countries, are diagnosed clinically because those false results. So I told you that means morbidity, mortality, loss of funds, and which to us is a big concern, is many of the small and the medium hospitals are breaking down because of that. We also collect preliminary results in the laboratory and in the clinical field. This is one of them. This is our high-risk group. It's an HIV-infected population. They were smokers, so dual reason for high risk for COVID. And we are located in Florida. Florida is one of the epicenters, the biggest spots here in the United States. And if you see, we were following 400 individuals, and of those 400 individuals, which has a good distribution across sociodemographics, they were tested in average 2 or 3 times per year. During that year, they report 34 that they were close contacts for other infected individuals. 10 of them say that they have clear symptoms, but the test was negative. And only 3 individuals were told that they were infected and they were followed. So if you see in this graphic, this was Florida at the time that we take that study. So it was 20% of the population was positive at that time. Obviously, we are talking about a high-risk population. We was expecting that we will have at least that 20% or even higher. So we really have, as I showed you before, the 1% with the PCR, and 4% of them were with symptoms. So we decided to go back and do antibodies, IgG antibodies, to be sure if those individuals were infected in the past year. And we find out that 50% of them, in addition to these 4 and 1, were really infected during that year with COVID. That really was better and more agree with the 20% that was going on Florida. With our test, we were able to detect 16% of them. So obviously the big question was how we were able to detect so many cases that the PCR and the antibodies were not detecting when they were doing it at repetition. And we went back with the animal models. We used the animal model to try to track our target and do both a low doses and a high doses to be sure that we can represent those with asymptomatic and clinically symptomatic cases. And we were able to demonstrate that our target for diagnosis has started being heavily produced at this level, at this day, approximately the 3rd day post-infection. But equally important, when we check for our target, we were able to demonstrate it through the entire time of the infection and the disease. Means we are not going to have these gaps or these gaps. So what we are trying to create right now is the 5th generation combo antigen antibody test that will further increase our accuracy. A difference of the other tests that needs to be done in the lab or needs to be done with swabs. This is a drop of blood. It will allow for early detection and will also give us the benefit of not testing gaps. A part of that, we're using multiple antigens, and that will allow to recognize vaccine versus current infection. This is the group. I need to give credit to the rest of the team that is helping me in these efforts. And thank you for your attention. And if there is any questions, open to that.
Thank you, Maria.